No. Hi, so welcome back to GLBIO. It's my great pleasure today to introduce Dr. Quaid Morris, who is the Canada CFAR AI Chair at the Donnelly Centre for Cellular and Biomolecular Research. I had to write down that, but thankfully it's all up in the slide here. Dr. Morris's research spans this really impressive set of domains. Since I've started following his work 15 plus years ago, he's been making these really key contributions across gene regulation, working on everything from understanding protein DNA, protein RNA specificity, through understanding the splicing code, through understanding how gene regulatory networks are structured and how that contributes to the genome to phenotype, the genotype to phenotype problem. Since then, and more recently, as we'll see today, I think, he's been expanding to understanding problems in precision medicine and in cancer genomics. There's three features that I really enjoy about Quaid's really impressive research. The first is how collaborative he is. So he has numerous collaborators in Toronto and elsewhere, not only on the wet lab side, but also, which is a bit rarer with other computationalists and machine learning experts. Secondly, and maybe because of that, his research always involves applying cutting-edge machine learning techniques to understanding biological problems. And then that's the third aspect of his research I really enjoy. When you read his papers, the biology is always at the forefront. Understanding the biology is really obviously his main driver. So rather than going on, please join me in welcoming Dr. Quaid Morris.
Hi, everyone. I'm Quaid. Hi. Thank you very much. It was a really nice introduction. I'm very excited to give this keynote. It's fun to be a GLBIO. I mean, we don't have to worry about like mean bi-coastal people here. We can all just be nice and have fun and do good science. And so, today I'm going to talk to you about, I have kind of a, it's a keynote, so you kind of get to do things a certain way. And so I'm going to call my talk Reading the Fossil Record of a Cancer. And like all good keynotes, we're going to start at, the founders of the field. I'm going to start with Plato. That's kind of a joke, sorry. Okay, so just to get into it, this is the key, what is the, all right, we're on, this is how it's going to work. Okay, yeah, so as Sean was saying, we do a lot of things. Cancer genomics is a bit of a newer field for us. Though there's a lot of people in my lab now working on this specific area, and that's going to be the work that I'm going to talk to you about today. We're still interested in gene regulation. A lot of our work has been involved with RNA binding proteins, fitting motifs to those, finding about the secondary structural impact on those motifs, and more broadly looking at trying to understand post-transcriptional regulation. And when you run kind of a machine learning and biology lab, you're always looking for new and interesting problems to try out new math with. And so right now we're looking increasingly at clinical data, some high content screening, and we've just gotten into metabolomics, though that's pretty new stuff. So going back to our cancer work, like all good research problems that come out of my lab, we get attracted into the problem with a math problem that we want to solve. And really, I'm focused on trying to solve interesting math problems, have an impact in biology, because it's a lot more interesting to say that, I spend my days solving math problems to cure cancer than it is to say I spend my days solving math problems because they're cool. And so, what that means is our perspective is a little bit different than most people's perspective when they come into a new field. And so we're going to talk about things from our perspective, and I'm hoping that'll give you kind of a new viewpoint into how to analyze cancer genomic data. What's going on? What are you asking?
Should I sort out the slide advance?
No, it's okay. It's because it's preview, and preview is not set up for slides. I've got it figured out now. The big question is, do you folks like this as a pointer or this as a pointer? Who likes number one? You just put your hands up. All right, who likes number two? Oh, the number ones have it, sorry. All right, so I want you to look at the cancer genome as a fossil record. And what do I mean by that? Well, what I mean is, I'm used to using this thing. I have to put that away somewhere. Okay, what I mean is that when you sequence a sample of a tumor, because the mutations that you see are, the somatic mutations that you detect in the tumor are persistent, those somatic mutations have accumulated during your entire lifetime and the lifetime of the tumor. Starting from when you were embryos, when you moved out into become an adult human, and ultimately one of your cells, when your cells acquired mutations that gave it selection advantages and started misbehaving and grew out eventually into what will become a tumor. And so the somatic mutations that people look at when they examine cancer genomics data, they divide broadly into two types. There are the driver mutations, and there are the passenger mutations. And so what are the driver mutations? Well, the driver mutations are the ones that drive cancer. They're rare, but when a cell acquires them, the cell acquires some sort of selective advantage. And those selective advantages, as after it accumulates 5 to 10 drivers, often that those are the number of drivers that you generally need to transform from a normal behaving healthy cell into what's going to become the founder of all the cancer cells in the tumor. And the clonal evolution theory of cancer posits and has a lot of evidence supporting this viewpoint now exists, that these drivers, what they do when they're acquired, they provide a selective advantage and they lead to either partial or full selective sweep that defines large lineages of cells. And so what happens is a cell gathers a driver mutation, it increases in frequency compared to other human cells. And both the driver and the passenger mutations, these are mutations that were already in the genetic background when that cell acquired a mutation, increase in frequency in the mutation frequency population. Okay, in contrast, there's the passenger mutations. So these are the ones that hitchhike to increase in frequency. They're common, meaning that there's about 100 to 1,000 of them for every driver. So when you sequence do whole genome sequencing of a tumor sample, a large tumor sample, on average, you detect about 5,000 mutations when you sequence to a depth of about 30 to 50. They're random because they don't provide any sort of functional advantage. And I put that in quotes here, and then they also hitchhike to increase the frequency. So the only reason that you see these somatic mutations when you sequence a tumor is because they are in some genetic background. And they are in some cell whose frequency got increased because that cell also contained a driver. Now, what I'm going to talk about today is looking at these patterns of mutations as a way to try to reconstruct the series of transformations or the changes that the cell underwent from being an adult human to becoming a tumor. And I'll be a bit clearer about what I mean by that in a second. And we're going to focus largely on the passenger mutations. And we're going to do that because they're not random. The passenger mutation, the type of passenger mutation that you see depends on the mutational stress that caused it. And where passenger mutation accumulates in the genome depends on, in some weird and complex way, on chromatin state, right? And so both the where and the type of mutation that you see when you look at passenger mutations are saying something about the state of the cell that those passenger mutations are accumulating in, right? And because these mutations are persistent, what you see at the end when you actually sequence a tumor is you see mutations that have been accumulating during different cell states as the cell was transforming from, in most cases, a differentiated cell into a cancer cell. Okay. So that's the premise we're going to work from today. And boom. Okay. And so here I've shown you a picture of like what's called the Vogelgram. This is one of the more well worked out genetic pathways for transformation from colonic stem cells into colorectal carcinomas. And there are multiple steps in this pathway. And you can see that each one of these steps is associated with some sort of driver mutation that's been gained that activates or deactivates certain pathways. And those gains of driver mutations are associated with phenotypic changes in the cells, right? And so then when you map the driver mutations onto this Vogelgram, you can see that the passenger mutations that are accumulating before the acquisition of the driver mutation
are accumulating in a specific cell background, right? And so if we could read them, if we could read out what the pattern of passenger mutations is telling us, what we have is a type of molecular phenotype for cells that we can no longer see, right? Because those cells are gone or transformed, but they're appearing in this background. Okay, and so here I'm showing two types of mutations. And so let's say here that what we're going to call the mother of all cancer cells is the most recent common ancestor of all cells in this carcinoma, and most of the tumors that you see have a single clonal origin, meaning that they came from one single cell. I'm going to call those the mutations that were in those cells. Those are going to be the clonal mutations, and the subclonal mutations. Those are going to be cells that are those are going to be mutations that were accumulated after this ancestor of all the cancer cells in the carcinoma, right? So mutations that were present in that cell to a first approximation are in the first cell are going to be present in all the cells in the carcinoma, and mutations that are present in mutations that are present in the subset of the cells can be distinguished from those that were clonal based on how frequent they are in the populations you sequence. Okay, does that make sense so far? All right. Okay, and so we're going to try to characterize these cancer populations by whole genome sequencing. So generally speaking, we have this is the context that we're going to be looking at. So we have what's called a bulk sample of the tumor, which contains a variety of cells that are some that have some shared genetic ancestry, but have distinct somatic mutations. And everything we talk about today, more or less, we're going to be looking at data from whole genome sequencing by short read DNA. Why this is important is we're going to be trying to construct phylogenies of cells. And generally speaking, when you construct a phylogeny, you have a whole genome, but we're not going to be able to have a whole genome because we're looking at population statistics of allele frequencies to construct these phylogenies. So we're going to have to use non-standard phylogenetic techniques to do that. And so what we're going to, the way in which we're going to represent the phylogeny is we're going to make what's called a clone tree reconstruction. And the reason we do that is because it's it's a better map to the data that you see. Because when you look at groups of mutations that have certain, that appear in different epochs, those mutations are going to be shared by subsets of the cells. And so each epoch corresponds to some sort of subtree within the phylogeny that you're going to be building. And so you can think of the clone tree as just a clade tree. It's a tree showing the relationships between different clades or subtrees of the phylogeny of the cancer cells that we can detect. Okay, and so today I'm going to talk to you first about how to identify these epochs that might be historical, and that's going to be subclonal reconstruction, and we've developed a variety of techniques to do this. And then I'm going to tell you about what we can do once we have identified these epochs, so inferring these historical states from the patterns of mutations. Okay. So when you do whole genome sequencing of tumors, you can detect multiple types of mutations. We're going to focus on the point mutations, or the single nucleotide variants, or the indels. And then we're going to quantify how common those mutations are within the tumor, the heterogeneous tumor population we're sequencing using their variant allele frequency. So you identify a mutation that's not present in the germline genome of the patient, and you count how many reads have that mutation, and that gives you an estimate of the allelic frequency of that mutation within the cell population. Okay, and so mutations that are present in every cancer cell should have an allele frequency under most cases of about 50%, because a lot of these somatic mutations are going to be heterozygous. Though there's 2 alleles per cell, and if one of them is a mutant allele, you expect 50% allele population. We're also going to be looking at copy number aberrations. So these are changes in the number of alleles of different loci. A very drastic one is a whole genome duplication. This occurs, as I'll show you, in about a third of cancers, so a third of primary tumors. But you also see gains of whole chromosomes, gains or losses of whole chromosomes or chromosome arms. We're not going to be looking at what are called structural variants. So some of the gains and losses are localized to small regions of a chromosome. For example, there are processes in cancer like chromothripsis, where like a single chromosome breaks up into a lot of pieces and then gets stitched back together in kind of weird ways. So you get loss and gain of parental copies of the given chromosome, and everything appears in kind of different orders. So you get large insertions and deletions and inversions. And those you can detect by identifying a breakpoint when you do the whole genome sequencing. They're very hard to detect. Methods and because they're hard to detect, they're hard to quantify the allele frequency in the population. So those estimates are going to be a little bit less reliable, and we're not going to be looking at those. Okay, so how do we group mutations into these historical epochs that might represent different cell states? Okay, so let's say these two cells represent two cells in this heterogeneous cancer population. I'm showing you they've undergone whole genome sequencing in some shared ancestor. So there's 4 chromosomal copies at the locus that I'm showing you, and by the boxes here, I'm indicating individual mutations. Now, of course, we don't see DNA in specific cells. We see DNA from a bulk tumor sample. That's the situation that we're going to be looking at. And so that involves doing a cell lysing. And so now we just have allele counts. So this green mutation is in about 50% of the alleles that we see in the population. The yellow one's on 25%, and these other ones are individually in one of the 8 alleles. Now, I posit that you can, based on these allele counts, you can actually reconstruct a lot about the history of this specific cancer. So first of all, you can define what's called an early clonal period that occurred before the genome doubling, assuming that you can call the fact that there's been a whole genome duplication here. And that this green mutation, because it's in copy number 2 times that of the yellow mutation, was present before the genome doubling. There's also a late clonal period where the ancestors of this cancer acquired further mutations until the most recent common ancestor cell. So any mutations acquired during the late clonal period are going to be present, in this case, in 2 copies. And then there's a subclonal period in which the individual mutations are going to be present at less than the clonal frequency. And so if you just look at the allele frequency of each one of the mutations, and you perform some transformation to it, so you can recover the estimated number of mutant alleles per cell, and then you just plot a histogram of the number of estimated mutant alleles per cell for each of the thousands of mutations that you detect. In this case, where there's been a whole genome doubling, you'll see a peak corresponding to approximately 2 mutations per cell, a peak corresponding to approximately 1 mutant allele per cell, and 1 or more peaks corresponding to subclonal populations. Right. And now simply by looking at the allele frequency, assuming that we can do all these transformations that I talked about, going from allele frequency to number of mutant alleles, we have now separated the mutations into 3 or 4 different groups that we can then individually analyze. Okay. Now, most of the time, some of the time you're able to detect this difference, but most of the time what you'll be looking within the subclonal epochs. So, and then you might get further resolution in this peak right here. And so a lot of what my lab has been working on over the past 5 years is looking at that individual peak, kind of expanding it out. And within that individual peak, you can see other peaks that correspond to subclones. And so subclones are clades of the cell phylogeny that have been caused by potentially some sort of selective sweep or some sort of drift process by which there's surprisingly large frequency of cells from a particular subtree. Okay, so just quickly going through this picture here, I'm defining we have this kind of clonal cell that I'm going to call A. It's got some set of mutations that it's acquired. It includes 1 or more drivers plus all the passenger mutations associated with that. And then there's 3 subclones representing clades of mutations or lineages of the tree that I don't know. I'm just not feeling it today. How is everybody feeling? I'm gonna have a little bit more of my breakfast, and let's kind of change this up so we have a little bit more fun. Okay, great. So you can see these subclones are arranged in some sort of a tree. Each one of these little diamonds corresponds to a set of mutations that, in this case, let's just say that the different clades have been driven by some sort of subclonal driver mutation. So there's some sort of partial sweep that increases the frequency, all the cells that contain, in this case, the C mutations, which could include one driver and multiple passengers, let's say. And so there we have a lineage of cells C that's descending from lineage of cells B. We have lineage of cells D that's descended from lineages of cell B. And then when we actually take the tumor sample, we're sampling mutations from 3 different subpopulations. The gray population, which hasn't the gray lineage, which hasn't disappeared. The yellow lineage, which is still there. The periwinkle lineage, which is still there. That color come out? Yeah. And even though the green lineage is, there's no cells that only contain the A and B cells, mutations, it's still being represented because it's present both in the C and D lineages. So now if we just take the varying allele frequency, do this magic transformation into the estimated number of alleles per cell, and then plot them, you know, what we would hope to see is hope to see 4 peaks corresponding to each one of the lineages. And there's one of the peaks at 1 mutant allele per cell because all the cells in this population are derived from this clone lineage. Okay. And then we can define these mutation sets, and you know, in this case, we can also derive something about the relationships between these lineages. Okay, so now, I'm sorry, arrow pointers. I like doing this better. Okay, all right. So, but I'll move it around so it's easy to see. Okay, great. So let's look at this. So we've got these. We've just plotted basically the varying allele frequency, the histogram of the varying allele frequencies of the, in this case, let's say thousands of mutations that we're detecting in this sample. And look, beautifully, we have 4 nice peaks here. Right? And so, and I'm telling you that each one of these 4 peaks corresponds to one of the lineages. The reason that they're not like delta functions is because there's some error in our estimate of the varying allele frequency because we're doing finite sampling of, of sequencing. Okay, so there's going to be a little bit of spread here. Now the problem is you can, one of the problems is you can see there's a lot of overlap between these peaks. Right? So if I took a mutation from this peak here and I'm like, well, which lineage is this? Is it an A and B? I think you'd have a lot of trouble telling me that. Right, because you would presume that this peak kind of bleeds over here and this peak kind of bleeds over here. So these mutations in the middle, we can't really assign them to lineages. And so we have some uncertainty about the mutation assignment. Furthermore, I don't know, is that 1 peak or 2? It kind of looks like 2, but you know, you could argue that it's 1 peak. So there's going to be also some uncertainty in the clustering of these peaks as well. So this whole thing about the whole subclonal reconstruction I told you about, it's not as easy as I tried to make it seem. Right. So let's first assume that we can actually get these peaks right, and then from that we can look at the mean and estimate the number of alleles per cell. Well, with that information, we can actually build out the relationship between these different clades. Right. So imagine you didn't see this, and someone just told you, well, there's a clade that's at 1 allele per cell, there's one that's 0.8, 1.5, 1.3. Well, we know that that there's going to be some cells that contain both A and B because A is in 100% of the cells and B is in 80% of the cells, right? And if there's some cells that contain both A and B, well, the AB cells, they could either derive from convergent evolution, so that 2 cells could have independently gained the same 100 mutations at the same time within the evolution of a single tumor, which seems unlikely, or the cells that contained A gained the B mutations. You can use the same types of arguments to say, well, then there's cells that contain A and C, therefore C came after A, and there's cells that contain B and C, so C came after B. You can't really use that argument to say what the relationship is between D and C. So D could have derived from B, or it could actually have derived from C. And we don't know that. We don't see this picture, all we see are these alleles. The cool thing is that we've actually been able to construct a lot of the relationship with the phylogenies of the cells. Some of them we just can't get, though. The second thing, oh, that suddenly worked. Okay. The other thing is, like I said, what if you get the clustering wrong? If you get the clustering wrong, you get the number of alleles per cell wrong. And now if you do these types of calculations that I showed here, we get a tree we're certain about, but is actually wrong. Okay. And then also we need to correct for varying allele frequency for copy number. So if we want to infer the number of mutant alleles per cell from varying allele frequency, we have to know how many reference alleles we should expect per cell and how many mutant alleles we should expect per cell. And cancer keeps changing the counts of the number of chromosomes and making these like new neochromosomes. And so this can be a bit messy.
Luckily, when you do whole genome sequencing, you can often reconstruct the copy number fairly accurately, even in a kind of a subclone thing. But in order to know the relationship between copy number and mutant alleles per cell, you actually need to know whether or not a copy number change is in the same cell that a mutant allele is in. Okay, this is complicated. And because of that, we developed a Bayesian method to do this. And the Bayesian methods, if you don't know about them, they rely on what's called a generative model, where we say, well, we could score this varying allele frequency histogram if we knew what the tree was and we knew what the clonal frequencies were for all the subclones. And so that gives us the score of the fit of a tree. And then also, we might be able to say something, we have some expectation about how that tree might look, or maybe we just want to say that we have some uniform probability over all possible clone trees. And then given a tree, we have constraints on what the frequency are. And so we can express those constraints as a probability distribution. So given a tree and a set of frequencies and the data that we observe, we can multiply all these numbers together to score a tree, right? And if we set these things up in the right way, they have probabilistic interpretations, which means that assuming that we're right or relatively close to what the real generation process might look like, we can actually say whether or not we're certain or uncertain that a specific tree fits the data. Right, and so, well, what do we have to do now? Well, we have to search over all possible trees and frequencies because we're not observing those directly to find ones that score these well. And then the trick to do that is called Markov chain Monte Carlo, and it's kind of complicated, and there's technical details that I don't need to tell you, but luckily we solve them, more or less. And so that's what our method called Philo-WGS does. Okay, I don't know what's going on here. Yeah.
Is the model in there in those formulas, does it model the copy number or is that somehow considered fake?
Yeah, great question. So it's assumed to be provided.
Yeah.
And when we made this model, it was like 5 years ago, and we read these papers that told us that the copy number reconstruction problem had been solved. So we assumed that our colleagues would provide us those copy numbers. Now we're not so sure that that's true. And certainly we think that there's a lot of ambiguity in calling subclonal copy number, especially from whole genome sequencing data, right? But here we are assuming that that problem is solved. And people are like working on that problem as we speak. Great. And so we did this, and like I said, this has been around for 5 years. We use it, other people use this method. You know, it works okay. Well, it's my method, so it works fantastic. And you should all use it. But it's pretty slow. And it's slow because we're doing this kind of search over this tree and frequency space. And if you know anything about phylogenetics, you know that like searching over the space of all possible trees is fraught with challenges. And we have sort of similar challenges when we do these types of searches. And so for some large problems, it can take a month for this to run. Like for like reasonable problems without a lot of copy number change, it can take a few hours, but we have seen it take a long time to run. And furthermore, about 2 years ago, you know, we got some collaborator, and the collaborator's like, hey, can you build us some trees? And we're like, great, we're going to build you some trees, give us your data. And they gave us their data, and it was great data. And I'll tell you a bit more about that data in a second. But it was like the variant allele frequencies are pretty accurate because it was sequenced really deeply. They had dozens of patient samples and based on those dozens of patient samples, there were like 15 or more subclones existing in the data. And it was so good, the data, you could just look at it and figure out what the tree was using the kind of rules that I'm going to tell you about in a second. And we found that our automated method on like what we think is the easiest possible version of this problem just failed completely. Didn't fail completely. Okay, let me take that back. It's my method. Didn't work as well as we had hoped. And so we went back and said, well, what is going on here? Like, you know, as the data gets better, I can't believe that the problem actually gets harder. And it wasn't only our method that didn't do that well. We tried a bunch of other people's methods too, and they didn't do well. They did worse than our method. And so
So that led us to on a new research agenda where we developed a program that we call Pear Tree. And so there's kind of a key insight here. And so I also just decided this morning that I was going to tell you about Pear Tree. And so I copied the slides from my my great grad student, Jeff Wintersinger. And so you can see he uses some sort of thing that's not Keynote. And I just got images. So you'll see what that looks like. Don't worry, he makes good slides. Okay, and so the key idea here is the reason that these trees were so easy to construct was that for any pair of mutations, we could tell what their phylogenetic relationship was. Just by looking at the data, we could tell if two mutations were ancestors or descendants of one another, or if they are on different branches of the phylogenetic tree. And your ability to do that gets easier as you have more data. And I'll show you that in a second. And so, and once you define what all the pairwise constraints are between mutations, you can actually just read out the tree. Because if I tell you the pairwise constraints between every pair of mutations in the tree, there's 1 or 0 trees that are consistent with that. And so that's the key idea of this algorithm. And because I only have 15 minutes left, I'm going to be a little bit more go through a bit quickly when I explain it. Okay. So let's go back to this picture that I showed you before, and let's assume we have kind of multiple samples. We have some sample from early cancer evolution. So we were collaborating with John Dick's lab, and they work on leukemia. So in blood cancers, you can get multiple samples of the cancer over time, and it's actually, there's a clinical reason to do that. So it's easy to get sort of multiple longitudinal sample data. So you sample a cancer population. This one's got 2 lineages being represented here, sampled again. Here there's 1, 2, 3, 4, 5 subpopulations and 7 lineages being represented, maybe then to sample a distant metastasis. And so you have these estimates of the cellular frequency of different lineages from 3 different samples, right? And so then you can just plot those cellular frequencies. And then, as I said, the contribution is to say that you can use those frequencies to find pairwise relationships between the mutations or lineages, and that's all you need to build the trees. Okay, and so there's basically 3 types of relationships. 2 mutations in the same lineage means that they're always present in the same cells. 1 mutation is ancestor of the other, meaning that all cells that contain B contain A, but not vice versa. This is vice versa. Or they're on different branches, meaning that they're mutually exclusive. A is in no cells that are in B and vice versa. Got it? And there's something we call the sum rule, and other people call it the pigeonhole principle, and I've already introduced this to you. So if you find any sample where 1 mutation is in nearly all the cells and another mutation is only in a subset of the cells, but the frequency of those 2 mutations add up to 100%, you know that this one is ancestor of the other one. And then there's something we call the crossing rule that basically says, well, if there's some sample where this mutation is in more cells and there's some sample where this mutation is more cells, well, neither of them could be ancestors to the other, so they're on different branches. Okay. If you got enough samples, eventually you'll see one of these constraints being satisfied. And so, because assuming you could do that, then you can define all these pairwise relationships and build out the tree. And for every tree, there's only 1 of these matrices, and 1 of these matrices either has 0 or 1 trees associated with it. Okay. Now the problem is hard, first of all, because it could be ambiguous. So you could not be able to apply either the sum rule or the crossing rule to some of the data. And also, though Jeff doesn't say it here, these are probabilistic inferences, right? So there's some probability that we're going to assign to the idea that one is ancestor of the other, or vice versa. Okay, and so this is what the data looks like. So we have dozens of samples, and this is whole genome. This is targeted sequencing from an initial whole exome sequencing. So we have a few dozen mutations, and the color here represents the cellular frequency. So dark red means it's in all the cells, and pink, which you can hardly see, but shows up nicely on my screen, of course, represents that it's in the subset of cells. And already here we can see these kind of sum relationships. So these mutations are present in all the cells, so they're clonal. Every cancer cell has these mutations that correspond to each one of these columns. There's a column over here
where the mutations, where this mutation is only present in some samples. There's this like weird column here where these mutations are present in samples that these mutations don't seem to be present in, so they're on different branches. Anyways, we went through this procedure, and with a little bit of automated help, we were able to kind of build out this kind of expert-derived clone tree that has a lot of structure. It's got 70 different subclones that we've defined. And it's kind of this nice, interesting looking tree. And this is data that was collected by Stephanie Dobson, who's an MD PhD student in John Dick's lab. She's fantastic. She generated data from thousands of PDX mice and collected data from like hundreds of patient samples over 14, sorry, not dozens of patient samples from 14 patients with AML. Okay. So it works. We applied Pear Tree to try to reconstruct this kind of expert-derived clone tree that we could see, and this measurement here is just some sort of reconstruction error, meaning the number of pairwise relationships we disagreed on. Pear Tree disagreed on like 4% of the pairwise relationships, right? And so, whereas our other algorithms, Philo Plus and Philo WGS, they agree on like 20% of the relationships. We're not showing data from another method we tried called Pastry and another method we tried called Lychee. They perform, I can see Leila's here, so Leila's here, so I'll talk about Pastry afterwards. Pastry either gets it completely correct or gets it completely wrong because it can't derive any trees, and there's a good explanation for that. And Lychee, we don't know yet, but we think it's going to be comparable with what we call Philo Plus. Okay. Any questions about that? Yeah.
You mentioned samples and patients. So what's the difference between the patients and samples?
Yeah, so you can have multiple samples from a patient. And then in this particular thing, what Stephanie did is she collected cells from a single patient with leukemia, where they took blood at diagnosis and then took blood, unfortunately, whenever the patients relapsed, and then took the cells, sequenced some of them, but also took some of those cells and implanted them in like immune-compromised mice. And then those mice grew out their own leukemias, and then she just sequenced those leukemias. So it's like these are samples from the same cancer, meaning they all share the same phylogeny, but the relative proportions of different subpopulations of the cancer differed. And those differences allowed us to infer what the relationships were. So we can't look directly at genotypes, but we can look at cellular frequencies. And those cellular frequencies can, in some case, tell us whether or not mutations are mutually exclusive or are always together in the same set of cells. Does that make sense? Okay, great. All right, summary so far. I've kind of focused more on methods. I've taken out the slides where I talk about biology. But when we run something like Philo WGS and we score prostate cancer biopsies in terms of whether or not they contain subclones, that we see like a twofold increase in risk of relapse when we control for all the other variables that are associated with risk of relapse in prostate cancer. This was recently published in Cell. And this is collaborative work with Paul Boutros and Rob Bristow. Also, we participated in a large cancer genome like TCA-like project where we analyze data collectively in a group of others, 800 other researchers for 2,500 whole genome sequence tumors. And we found that subclones were ubiquitous and whole genome frequency duplication is frequent. And I'm going to talk a little bit more about the data from these 2,500 tumors in this next section. Okay, yeah.
How does your model handle tumor purity?
It handles it just fine. So tumor purity is just saying, well, instead of like the clonal mutations being present in 100% of the cells, they're present in, let's say, 70% of the cells that the tumor has 70% purity. Where you have to be concerned about tumor purity is for models to work well, you need to have accurate measurements of the variant allele frequencies. And as the tumor purity decreases, you're getting fewer and fewer reads from subclones. So your estimates of the variant allele frequency decrease in their accuracy. Yeah. but it pretty much, like, you can read off the tumor purity when you do the reconstruction, because there's going to be one clonal population, unless you have it in some mode where you imagine that there might be like multiple tumors of independent origins represent the same samples. Any other questions? Yeah.
I'm curious about the variation in the variant allele reconstruction. Nominally is sort of binomial, but then you'll have some. It seems like extra binomial variation could really affect your conclusion. And I just wondered what your.
Yeah. So this is not a problem I think is like well solved in the field yet, and we've been pretty lazy about getting around to solving it because we think there's other interesting problems. Yes, there's probably some over-dispersion. Models that don't include over-dispersion seem to work pretty well under most circumstances. The problem is, if there's over-dispersion, you need to know how much over-dispersion there is. And so figuring out how much over-dispersion there is it requires you to make some assumption about how many subclones there are represented in the population. So it's kind of a chicken and egg problem. And so we have some ideas about how to do that, which we haven't implemented yet, but other people haven't implemented them yet either. But it's an open problem in the field, I would say. Yeah. Okay. All right, so now let's talk a little bit of biology. We want to say something about inferring cell states. And who's here has heard about mutation signatures? Wow, that's a pretty good crowd, actually. It's about half of you. I guess I put cancer in my title so that you would come. I've given this talk to when people haven't heard about mutation signatures. It just blows their mind. So what's a mutation signature for those of you who haven't heard about it? Well, you can classify mutations, in this case, into one of 96 different groups, depending upon what type of mutation it is, from, for example, a C to an A, and the genomic context of the mutation, so the 5' base and the 3' base of that mutation. There's 6 different types of mutation because you don't know which of the 2 strands the mutation occurred on. There's 16 contexts, so there's 96 different types of mutations. And then if you just plot the count of each of these 96 mutations detected in a single tumor, you get something that we call a mutation spectrum. And if you look across different tumor types, the mutation spectrums are quite different. And actually, if you've been looking at these things long enough, you can read this. It's kind of like palmistry, right? And so this is the lifeline here, these 4 peaks. And they correspond, this is controversial, but they correspond more or less to the age of the person that the tumor was taken from, because they represent mutations that seem to occur at a relatively consistent rate. So I can say that this is an older woman that this breast tumor was taken from, probably. And then these mutations over here, you can see in the lung cancer, there's a lot more of these red mutations than there are in the breast tumor. Any guesses for what the red mutations represent? Smoking, right? And so mutagens that are present in tobacco or in cigarettes cause these CC to AA mutations. And so in smokers, that manifests as an appearance of other things. Here we can see that there's been some activity of another process, cellular process called ApoE, which generates a very specific type of mutation. Okay, so like we're saying things about cells and we're saying things about the mutational processes just by reading off these spectra. And so then this beautiful work that was done about 6 or 7 years ago in Peter Campbell and Mike Stratton's lab by a number of people, including Ludmila Alexandrov, David Wedge, Serena Nick Zainul. What they did is they looked at these mutational spectra across like thousands of different tumors, and they identified patterns of co-occurring mutations using a technique called non-negative matrix factorization. And initially, they identified 30 different patterns, and those are shown here. And those are shown just in terms of like the frequencies of different mutations associated with different patterns. And simply by looking at these, either the patterns of mutations themselves or the types of mutations, the tumors these mutations appeared in, they were immediately able to associate each one of these mutational signatures, what these called, with specific mutational processes, okay? And the cool thing here is also there's a whole bunch of other signatures that we don't know anything about, right? And so we have like this bioinformatic analysis that's not only been validated in some sense, but also gives us a lot of interesting things to look at, okay? And so you can take the mutational spectrum of a given tumor, and then you can just break it down into what they called exposures, and that's just the proportion of mutations from each one of these signatures, okay? And that can give you some idea of like, well, where did the mutations come from in a specific tumor? And this is helpful. I mean, you know,
someone who has a lung tumor and they smoked, obviously they're going to have a lot of mutations from smoking. That doesn't help anybody. But some of these signatures correspond to internal DNA damage repair processes that were either defective or overloaded. And in some cases, knowing that can affect choice of treatment. Okay. So what we wanted to do, because we're interested in how tumors evolve, is we wanted to add, or what Yulia in my lab wanted to actually wanted to add a time component to this. And so, you know, the easy thing to do is that largely we, what I've shown you in the past part of this talk is to show that you can group mutations into different epochs, or in this case, what Yulia just did is she just sorted them by the average number of mutant alleles per cancer cell, and then you can just analyze each of these epochs separately in order to estimate the exposures. And then if you just plot this in some sort of timeline representation, you can see this beautiful change in the sources of mutation as the tumor evolves, right? And in this case is a very classic signature exposure trajectory from a CLL sample, where during the initiation or even before cancer, there's this accumulation of mutations in the B cell and the lymph node in a process called somatic hypermutation that gradually disappears. Those mutations gradually disappear as the cancer evolves. Okay, so now people have been doing something like this for a while before that by clustering the mutations as I showed you before, and then analyzing each of those clusters separately. But what we found when we looked at the data from the whole genome sequencing is that there's a lot of uncertainty in what those clusters are, right? And because there's these overlaps in the regions of different clones, and also because there's uncertainty in the exact number of subclones that are represented, what she did instead is she just reposed this as a segmentation problem, where she put all the mutations on this specific timeline, sorted by average number of mutant alleles per cancer cell. And we used a technique called PELT, so pruned exact linear time segmentation. Whoa. I thought I had more time than that. I guess I'm done. All right. And so what this does is it just segments this time lapse into different regions that correspond to different exposures. So it's like calling these, it's like doing the subclonal reconstruction, but we don't need to say specific things about clones. We only call differences when there's changes in mutation exposure. Okay, and then you can plot this over changes over a specific population. The important thing here, because I'm out of time, is just that these little wedges here tell you which processes decrease, which processes increase. The opacity of the wedge tells you and what proportion of the tumors of this type they increase and decrease, and the size of the wedge tells you how much they increase or decrease. So in CLLs, you often see signature 9 go down and signature 5 come up. And so as part of this PCOG project that I told you about, where there's 800 of us jointly analyzing data from 2,500 tumors, this is the distribution of different donor types. Along with Stefan Dentro, who's a student in Peter Van Loo's lab, and David Wedges' lab, we analyze these data, and we're able to build these kind of wedge diagrams for a bunch of different tumor types. The key insight here is they all look different. There are very distinct patterns, not only mutational signatures, but also mutational signature change among all these different tumor types. Also, it gives us a way of saying something interesting about specific mutation signatures. So for example, here, you can see mutation signature 17 always goes down, and that's indicative, or that's very similar of others, mutation signatures associated with mutations that occur due to carcinogens, right? They're important early on, and they gradually disappear, okay? And so the last thing that I wanted to tell you about, and so who's seen this plot before? There's like one person in the audience. Okay, this is really fun. Okay, the black line here, this is, so the plot here represents chromosome 2. And the black line here represents mutation density in melanomas. And these are a specific type of mutation, which is kind of important. As the C to T mutation, these are mutations that are caused by UV light. The blue line here represents chromatin inaccessibility. So it's basically, the blue line represents the degree to which chromatin is closed or inaccessible, as measured by DNase 1, and melanocytes, which is the assumed cell type of origin for melanomas. You can see here that the black line and the blue line are almost perfectly over top of each other. This is a figure. I think this figure alone could be a Nature paper, but it's a figure from this great paper from Shamil Sunayev's lab. Paz Polak now has his own lab in Mount Sinai. But what this means is, at least these types of mutations, they're accumulating in areas of closed chromatin. Now, in this paper and in subsequent papers, they then went on, basically, like took tumors of a given type, plotted the mutational density, and used that as a way of looking at data from like the epigenomic roadmap to identify potential cell types of origin. And they could do that because probably about half of the mutations that you see in the tumor are accumulating before the cell is doing the transformation, right? And why this is so exciting is it makes a direct connection between mutation location and cell state, as cell state is represented by open and closed chromatin, right? I mean, I think this is really cool. I just blew my mind when I saw this. And so what we were trying to do then is saying, okay, well, what can we say about individual tumors, right? So we have an individual tumor, like, you know, the way in which they did this analysis is they broke up the chromosome into 1 megabase bins. So there's 3,000 of them. And I told you individual tumors on average have 5,000 of these bins. How much can we say about the chromatin state of mutations, or even of cells, either the tumor cell itself or some sort of historical cell, just by looking at the spatial patterns of the mutations? Does that make sense? And so, you know, we looked at this for a while, and then we sort of took a step back and asked ourselves, I think, a different question, which is like, well, let's say you just gave me the locations of every mutation that you see in a tumor. Could I tell you what the cell type or what the tumor type was? And this is a question both for us. This is a question that that actually has some clinical value, because about 3% of cancers are what are called cancers of unknown primary, where the metastasis is found but not the primary. And so in this case, in some cases, the treatment of the primaries depends on, treatment of the cancer depends on what the primary was. So we could actually try to solve this cancer of unknown primary thing, but also ask this question: how much information is there in the spatial patterns of the mutations that you see? Okay, and so in order to do that, Gurnita, in collaboration with Lincoln Stein's lab and Wei Zhao, and actually Paz Polak, came up with these different types of features. So first there was the spatial distribution features, and this is just counts of mutations in individual 1 megabase bins. And then there's the SNP type feature, which is the mutation spectrum across these 96 different mutation types. Okay. And then he trained a deep neural net using like standard techniques in order to try to estimate the how well we could recover the type of tumor as classified by pathologists from 1 of 24 different states from simply these two types of features. And so before we joined this project, Wei Zhao was already trying to answer this question, but he was looking at the value of individual features, so like SNP type, SNP distribution, and the presence or absence of very specific driver mutations. He was looking at this with the random forest. And this is a F1 score, which is, you can think of it just as a kind of a lower bound on accuracy. It's the harmonic mean of precision and recall. And what you can see with Wade discovered, and this is pretty amazing, is that just the SNP type alone, or the SNP distribution, allows you to classify tumor type at about 75% accuracy, whereas the driver mutations, which you would have expected would have been particularly important, your accuracy hovered around 40%. And the reason this is, well, one of the reasons is about 25% of the tumors we looked at didn't have obvious drivers. And some of the driver mutations everybody thinks about, but like P53, they're not tumor type specific. There's like a couple driver mutations that are very tumor type specific and they're pathognomonic, but in those and you can see, you know, there are some points up here where the points represent individual cancers. you do pretty well on those, but in general, you don't do, you can do better across multiple cancers just by looking at these different types. And in these cases, which I'm showing over here, the passenger mutations themselves are already providing the information that you need to classify the tumor type. And so that's what this figure over here is. So Gurneet trained a deep neural network on these two types of features. This is the performance level he got, which was nearly perfect. When he added in the drivers, there was no change in performance level, so it was redundant. And the drivers themselves got performance about equivalent to what Wei was getting with the random forest, right? So the passenger mutations are telling you everything you need, at least in the whole genome sequencing thing, about the tumor type. And they contain a lot of information. And so this is the confusion matrix that gives you the overall accuracy. Previous accuracies on kind of the sort of the similar classification problems are given here. It depends on the number of tumor types. RNA does reasonably well, as you might expect, because it's a direct molecular phenotype, but it's hard to get. And we're doing nearly as well, in fact, better in RNA in this case. And then the types of mistakes that we're making from the confusion matrix, for example, are cancer types that are difficult to distinguish, like stomach and esophageal cancer. Different types of B-cell cancers are sometimes hard to distinguish because they have similar cells of origin. So he trained this on these 2,500 tumors from the PCOG project. And then around the same time, we saw this paper in bioRxiv where someone did whole genome sequencing on metastatic samples of metastatic tumors. And then just out of the box, Lincoln immediately contacted this group in the Netherlands and said, hey, we have this classifier, you want to collaborate? And they sent us their data. And just out of the box, I'll show you how well it did. It got an overall accuracy of 87%. Now the tumor types were, luckily they had a lot of breasts, which we do particularly well on. But the clinician accuracy at this task is about 49%. And this is a classifier that's just trained on primaries, right? So we don't have to worry about changes in the microenvironment that might occur within the cell due to being in a different tissue, because the mutations themselves are consistent. Okay, great. So this is what I told you about. Thank you for your indulgence and letting me go a little bit over time. So where are we now? Well, we can define these epochs. We can sometimes define clonal epochs. We can infer cell states from the mutational patterns, and this analysis can be done at a kind of an epoch level. We're getting towards being able to say things about chromatin states in different epochs. The problem is of course, we just don't have enough mutations, and individual mutations aren't giving us enough information. We've looked at this a little bit. So this like uniform genome segmentation, we can measure the amount of information that you get, and it takes about 27 mutations in order to give us a bit of information about tumor type. We can resegment the genome optimally doing something I call gerrymandering. I think maybe in Wisconsin you know something about it, I'm not sure. And if we do that, we can nearly double the amount of information that we get from the mutations. So what do we need? We need some sort of location and type signatures, because not only does the relationship between chromatin state, genomic location depends a lot on the mutational processes causing the signatures. And those types of signatures, that type of analysis, that's what people are thinking about and trying to work on right now. And if we had something like that, we would get a lot more information, presumably about chromatin state, by just inverting those models. I'd like to acknowledge the people who did the work that I presented. So everybody from my lab who did the work is in bold, and you've seen various members on the slides. These are our collaborators from the PRECOG project. Here I've listed the names of all the senior and first authors of the papers. You can see it's a big thing. We have a lot, we benefit a lot from collaborations with computational researchers, the Ontario Institute of Cancer Research, and experimental researchers. Also, we collaborate with Paz, we collaborate with John on the data that I showed you about. These were the lovely people in the Netherlands who shared their data pre-publication with us, and they're now authors of our paper. And these we were funded by these bodies in this work. Through NVIDIA, we were funded by the Compute to Cure grant. This was my first cancer grant ever, was from NVIDIA. And thank you. I hope I have a few minutes for questions. Thank you very much.
Short questions.
Yeah.
So I was wondering on the last part of the work, can you get at deconvolution of cell states, like to say, were the mutations primarily accumulating while your tumor was in a stem-like state versus non-stem-like?
That's what we want to do. We haven't gotten our mind around how to do it. We've tried a couple things, and I can say that they mostly don't work. So we're getting there. I think the key is building a really good mutation rate model that depends on chromatin state, might depend on like local features. And so we're looking in the literature. People have some of these models, but I think once that model gets really good, then you can say, look, we've seen a change in the mutation density in these parts of the genomes. Is there a way in which we can explain that change in density in terms of change of a specific chromatin mark? That's the way we're approaching it right now. Yeah.
Any additional questions? We had a couple during the talk, I guess.
Yeah. Well, thanks everyone. You're really nice.
So before you go.